was a really successful thing yesterday. Okay, so it's time to start. We're a bit tight in time because there's no break after the session, so we should be starting on time. As I said, thank you for coming so early morning, and welcome. And we have 3 wonderful talks today. And just without any delay, I will just introduce Mohammad Bakhtiari, if I pronounce more or less, that is going to tell us about federated and privacy-aware medical differential gene expression. The floor is yours.
Hi, I'm Mohammed. I'm part of Fisher Cloud core development team in University of Hamburg. We are working on a federated learning platform, and one of our applications is FLEMA. FLEMA is a federated version of Lima, which is a popular tool for gene expression analysis. We know that genes can be significantly over or under expressed in different groups of samples, and gene expressions can show the role of gene in different cells and tissues. But one of the problems with Lima application is that it asks all the data to be gathered in a centralized fashion in one centralized custody, and that be problematic with GDPR law. GDPR was enacted in 2018, and it's about respecting the patient's privacy by not compromising the data of the patients. And we know that genomic data is identifying, and also genotypes can be inferred from patients' derived gene expressions profile. And we know for sure that gene expression is sensitive, and also patient-derived omics data are also sensitive. With all these sensitive issues regarding the gene expression, we find out that at one point we need to have more data for training and analysis using Lima application, and on the other hand, we can't have it in one centralized custody. So what can be a solution is federated learning. In federated learning, in contrast to centralized training, instead of gathering all the data at one centralized custody and doing the analysis there, to fit a model on the data and train the model to provide parameters. In this simple case, which is linear regression, the parameters will be y-intercepts and slope. In federated learning, we leave the data at data sources. We deploy models in different hospitals. There, they can fit the model on their own local data and communicate some parameters to a centralized custody. Then that centralized custody aggregates the parameters, which can be simply averaging the parameters, and send back the aggregated parameters to clients. Now we can use federated learning to come up with a federated solution for Lima, but there are many platforms to use, and the one that we suggest is Fisher Cloud, which is a cool platform that addresses a wide range of issues from getting the concept of patients at hospitals to use their data in collaboration with other institutions in a federated fashion, to the other side of the story, which is about federated app developers who wants to develop federated application and provide it to hospitals to use it. We like to encourage you to visit our website, fishercloud.ai. There you can find tons of applications that we developed, and there are really cool videos and links that can help you find out how you can use the platform, either as app developers or as institutions who wants to collaborate with each other to train the model in federated fashion. Sorry. Federated learning tries to keep the data at data source, but It also needs some enhancing mechanisms to ensure that whilst the model is trained on the data, the data cannot be extracted from the trained model, or once we are transferring the parameters from clients to to centralized custody, the raw parameters will not be revealed to anyone. So there are some privacy enhancing mechanisms like homomorphic encryption, differential privacy, and secure multi-party computation. In FLIMA, we used SMPC, which is also available in the platform for every application. In SMPC, clients, instead of passing raw parameters, they generate some random noise and add the noise to the parameters and share the noisy parameters with one computational party and share the noise with another computational party. Practically, we can show that once we aggregate all the noises and noisy parameters, the noises will be canceled and we will get the aggregated parameters. In this process, we will not reveal the raw parameters of clients. Here we compared FLIMA with Lima. Lima is a centralized application which asks all the data to be gathered in one centralized custody, then apply the analysis to provide the results. On the other hand, FLIMA is federated and leaves the data at data sources, but reach the same results as centralized analysis without asking to have the data in one place. Okay, how you can use FLIMA application? You can go to our website. We provided a workflow, FLIMA workflow, which includes FLIMA app and a really cool cluster visualization app that provides some useful plots like Volcanopola. You can you can start a project, create a project and assemble a workflow or instantiate this workflow template. Ask other participants or hospitals to join in a federated collaboration with you, and then start the project and run FLIMA. FLIMA gets the same input as Lima, which includes gene expression and a matrix of confounders, in this case, which is related to breast cancer dataset. We have lumbar and basal subtypes and some other confounders, and this is the input data. In one perspective, FLIMA is some extension of Lima-Wum, so it includes at some point removing the weakly expressed genes, using upper quartile normalization to normalize data, removing mean variance trends, and finally fitting weighted linear regression to estimate the p-values. For every app in Feature Cloud, without any extra effort, you can have a state diagram that shows different states that you should execute in a federated fashion. Each state can be run by a coordinator or participants. We have one coordinator in the platform. One of the users will be coordinator, who is responsible to gather the data, and others will be merely participants, and each state can be run by either participants or coordinator, or both of them. Let's take a close look at this state diagram. At first, we filter on common genes across clients. Then we find the median cutoff of local data sets, and based on the cutoff, we filter weakly expressed genes. And finally, in the last stage of pre-processing phase, we normalize data using upper quartile normalization. Now that the data is normalized, we want to fit a weighted linear regression model, but we don't have covariance matrix, and for that case, we first fit a simple linear regression, then try to estimate the covariance matrix and remove the mean variance trend. And now that the matrix is available, we can fit a weighted linear regression, and finally find the p-values and provide the analysis. The output will be something like this, a data frame that provides you adjusted p-values, fold changes, etc. And also you can have a plot that shows differentially expressed genes in contrast to low expressed genes. For evaluation part, we evaluated FLIMA on two data sets. One of them is breast cancer data set and another is GTX16. We compared FLIMA against meta-analysis approaches because FLIMA was the first federated application. And we also considered the effect of imbalanced scenarios and batch effect on the performance of FLIMA. For the imbalance case, we considered different factors. One of them is cohort size, and we came up with three scenarios. Imbalance scenario, the cohort size will be approximately equal across clients, but in mildly imbalanced scenario, the cohort size will be double. In a strongly imbalanced, it will be tripled. To show that the FLIMA is robust against imbalance scenarios, first we compared LIMA-VUM, which is a centralized analysis, against meta-analysis, and FLIMA as a distributed analysis. If we compare the estimated adjusted p-values, if they are approximately the same as centralized, we can notice them in this orthogonal line. And you can see that FLIMA and Fisher and Stauffer almost provide the same values. And for balanced data set, but when it comes to imbalanced data set, we have FLIMA that keeps providing same p-values as centralized analysis, but here Fisher and Stauffer fails and tend to underestimate the p-values. To address the batch effect issue, we considered data from three different sources, while each data origin includes three different subtypes of breast cancer data. And for addressing the batch effect in FLIMA, we modeled cohorts as one of the confounders, and for LIMA part, we removed the batch effect using combat and then applied the LIMA on the data. In this picture, we compare predicted to fold changes against the LIMA for FLIMA, and we can see that the predicted values are 99% correlated. They have 99% PSI correlation coefficient. And by comparing the minus like 10 of adjusted p-values, we see that also there are 97% correlated. In summary, we come up with FLIMA, which is a privacy-aware federated tool, and it's robust to imbalanced data sets and also robust to batch effect. FLIMA is available in our website, featurecloud.ai/app/FLIMA. I recommend you can go and check the app. It's publicly available in the GitHub also, and it's fully documented, and you can use it. Thanks for your attention, and I will be thankful.
Thank you. Thank you, Mohammed, for just doing in perfect time. We have time for a couple of questions. Awake people in the room. There's a question, so it's my turn to ask things. I have 2 million questions on the way these previously were techniques, but perhaps my first concern is you're using linear regression, where actually combine the parameters is straightforward, I would say. How difficult can be the model to could apply this kind of approaches?
In general, in Future Cloud platform, how complicated?
In general, my question is more general.
Yeah.
So how, because people is doing deep learning and using privacy-aware techniques in deep learning, but I don't think it's that easy to just to combine the results as in linear regression. What do you think?
Yes, Feature Cloud supports different application. Currently, we have a deep learning application for medical imaging analysis. Soon, we will have a deep learning application for gene expression, for single-cell RNA sequencing data, for imputation, for cell type classification. Many deep learning application will be soon implemented, but now we have a general purpose deep learning application for medical imaging in Feature Cloud, and it supports wide range
That
of
works.
applications.
That's nice to know, because everybody's trying to do that, and everybody's asking people doing infrastructure to do that. So it's a good point. Is there a question there?
Yep.
Wonderful.
Good morning. Thanks, and nice solution for the federated architecture. Could you go back a couple of slides to the batch effect? Yeah, here. No.
Sure.
When you're comparing the
Yes.
results, yeah. So I was wondering why you compare FLIMA with the model adding the batch as a confounder against Lima using Combat, because those are pretty different. Or maybe I didn't understand something.
I can explain. Combat is also a centralized approach, and if we want to apply Combat on the data, we should come up with a federated version of Combat, which was published after FLIMA was published, or I guess it's under review. We are working on federated Combat also. At the time, we didn't have the federated version of Combat, so we wanted to address the batch effect issue inside FLIMA, so we came up with a solution of modeling the cohorts of the samples. Each sample belongs to each cohort as one of the confounder in the data set. And to be fair, we applied Combat on centralized data, and then applied Lima on the results. Please.
So, but did you also try to compare against this centralized model using the same model adding the confounder? Did you correlate those results? Which, well, for me, this is very interesting, but still lacking one comparison.
Actually, I guess we didn't compare it, as far as I remember, we didn't compare it with the centralized version that uses cohort as a confounder. But we know that Combat is a really popular app for batch effect removal, and we trust that once we use Combat, probably there will be not that much of batch effect to address.
Okay, I guess they also correlate pretty well, but would be nice.
Sure.
Okay.
Thank you.
Thank you very much.
Thank you.
Anyone else? More questions?
I'm just wondering, very stupid question, how privacy and friendly is RNA sequencing data, expression data? How predictive it is for genetics? I know about the EQTLs, but in my experience, the effect sizes are very small. So has there anyone tried to predict genomic profiles from RNA expression?
Yes, there are some applications in that regard. I guess you can check these papers. They talked about this issue, this Gimer's paper 2013, and also this paper. These two papers talked about generally the sensitivity of data and the fact that genomic data is identifying, but these are not the latest one. As you can see, probably there are newer papers that address this issue.
Actually, this was one of my questions as well. But should be really difficult to do that, or it's worth to try to identify people this way?
Yes, but because the cost is really high because of GDPR law, it makes this case, and the hospitals or institutions who has some data are really careful to share their data with other hospitals or
Yeah, but
any researcher.
I think there's a bit of paranoia there because actually the GPR says that it should be reasonable, easy to identify people to actually go that far. But it's fine. So thank you very much. I think there's no more questions. No, of course. No, nothing on the chat. Thank you, Mohammed, and I will just introduce speaker. Catherine Hayes is going to talk about sugars, glycan proteins, interaction motifs.
Hello, hi. My name is Catherine, and I'm from the PIG group in the Swiss Institute of Bioinformatics based in the University of Geneva. And I'm going to speak about a semantic-based annotation method for glycan-protein interactions. So a little bit of the biology behind it. A glycoprotein is any protein that has a carbohydrate moiety as a post-translational modification. So in this, I'm just showing a membrane protein here, but it can be secreted or soluble as well. So this is the representation of a carbohydrate moiety. And on what is presented to the outside world is an epitope or some small substructure of the carbohydrate that's recognized by a glycan binding protein. So in the Glycomics at ECCB platform, of which I'm part of, we have a couple of resources that I'm going to speak about. And the first one is GlyConnect. And GlyConnect is a curated database of glycan structures and the metadata that surrounds them. So, for example, the protein or the tissue that it's found in or the disease state that it might be implicated in. The other resource is Unilectin 3D, which again is a curated database. It also contains some predicted structures as well, but mainly curated 3D structures of lectins. And lectins are a particular type of glycan binding protein, in that they have a non-catalytic binding domain and they reversibly bind carbohydrates. They're involved in, for example, in the immune response. So the link between these two databases is the epitope or the determinant at the outside of the glycan that is presented by the glycoprotein and recognized by a glycan binding protein. So while we have these two resources, there's no automatic link between them, and biology doesn't differentiate between the two of them, so why should we? So we were looking at trying to identify an automatic way of linking the two. So this is an image of spike protein, again. It's from the front cover of the Essentials of Glycobiology, which was published last May, the 4th edition. It's available on the NCBI bookshelf. And this is a really nice image because it actually shows that the surface of the spike protein is covered with carbohydrates, okay? So if I was interested in how I might pull out these carbohydrates, I could go to Unilectin and see whether there was any kind of epitope in common that would allow me to pull out, we'll say, for example, the glycans that are on the surface of the COVID spike protein. So while traditionally, if I wanted to do this, I'd go to UniProt, I'd look up the sequence of the COVID spike protein, I'd scroll down to the post-translational modification section, I'd see there links to GlyConnect, which is our database. I'd go to GlyConnect, I'd see a list of structures, and you can see here the representation of the glycans is kind of a cartoon format. I'll speak about that a little bit later. But the green circle is a mannose residue. So I'd realized there's a lot of mannose, or there seems to be presented on the surface of the spike protein. I could go across to Unilectin and using the glycan search button, I could search for lectins that might be interesting for that. And indeed, you find mannose binding lectins. Okay. So the bottleneck in all of this is actually the epitope. So it's not the actual structure, but what's presented by the glycan. So we decided to look into trying to make a semi-automated substructure search to link these two resources. So a little bit of the chemistry. This is what people are very commonly see as a carbohydrate. So there's a lot of information in this chemical structure. This is the name of that it's a tetrasaccharide, so it's a 4 monosaccharide unit. I'm not even going to try and say that. So here we have the IUPAC kind of condensed format that a lot of glycobiologists would be used to. Thankfully, a few years ago, glycobiologists came together and decided on a kind of a standard nomenclature. So that's where this cartoon comes into play. This cartoon encodes all of this information between the colors and the shapes. So all the shapes are one isomer, a type of a monosaccharide, and the colors indicate the which isomer it is. And this is called the symbol nomenclature for glycans. And we also have linkage information there. So, again, if you look at the chemical structure, it's represented by a string format, which is SMILES. Our SNFG format is represented by this glyco-CT string format, which lists the residues and the linkages that are involved in those residues. So we were trying to come up with another format that we might be able to do substructure searching. But you might say, why? Why do we need another format? Because this structure here is an ideal structure. It has all of the linkage information encoded. But the reality is that if I look at this structure here, all of these structures have the same composition. They have the same molecular weight. Except this has all the linkage information and these do not. And this is the reality of glycan structure determination, because there is a combination of techniques used, mass spectrometry, HPLC, digestions. So if I go back to my epitope, if we think of this epitope here, lactosamine, in the box you can see the lactosamine units in all of these structures. So for example, here, it's a fragment. We know there's a lactosamine, but we don't know where it is on the structure. Here, we don't know what here we don't know what isomers are present, and so on. And worse than this, if I look at the glyco-CT format behind this, in bold, you see the lactosamine unit. So if I was to try and search for a lactosamine unit among all these different types of formats, it would be quite a complicated task. So we needed a way of creating a representation of the structure that allowed us to look for patterns. And it had to be easy. So we actually investigated RDF technologies, and we created an ontology called GlyStream, and it's based on a tree model. So it models the glycan structure as a tree. Now, we had previously created a model that included all the information, but we realized we needed to make it simpler. So we came up with this ontology. So we have over 5,000 structures. We transformed them into triples, and they're held in a GraphDB triple store. And we have a Sparkle endpoint that can be queried. And we're able to do federated queries because in GlyConnect itself, we also have an ontology underneath the metadata. And from this, we have IDs, and we can also query UniProt, for example, or NextProt. So if I look at my 3 structure formats that I have now, we have down here the SNFG cartoon. Behind that, we have the string format, and this is transformed into this kind of graph model here. So the first structure, for example, is represented here, here, and by this object. So the entire structure is represented by the residue list and the linkage list in the glyco-CT, by this part of the cartoon, and all these different objects here. But the most important part was that the undefined section, or this fragment here, for example, is represented by its own type of object, which is still linked to the entire structure. So we have a number of uses for this GlyStream. First of all, in-house, we used it to validate a lot of our structures that we have. And then we thought about experimentalists and how they might use it, and programmatic access. The programmatic access at the moment is by the Sparkle endpoint. But we thought about experimentalists, and if they want to use this as a substructure search, they're not going to learn Sparkle because it's not so nice. So we needed a method to automatically generate these substructure searches that our users might be interested in. So we took the algorithm that we used to create the triples. We adjusted it slightly to account for the missing information, and the output is a well-formatted Sparkle query. So basically glyco-CT to a Sparkle query, and we call this glyco-QL. So for example, if I go back to my COVID and I look at all of this and I see actually there's loads of different sites on the surface of the COVID spike protein that potentially have carbohydrates. And if I want to look at those, I can go to GlyConnect. We have a little subset of our data that is specifically for COVID. There's over 200 glycan structures. There's a list of them there, so you can browse here and see these. But if I want to look at them overall, I don't want to look at a list of 200 structures. So what we did was I took the over 200 structures and I looked at them and I created patterns that described the 5 classes. So these here are, I won't go into it now, but these are different classes of N-linked carbohydrates. So I created these 5 patterns, and using GlycoQL, I have so this is a pattern here, for example, for oligomannose structures. This is the glyco-CT behind it, and this is the GlycoQL translated glyco-CT. So this is what Sparkle looks like, okay? So this is a Sparkle query. So you don't want to be writing this manually, okay? So this is what GlycoQL does. It takes a structure like this and creates your Sparkle query. And what we were able to do then is look at the amount of different structures that are found for each type of pattern. Okay, so this is quite nice. You can see here we have complex neutral and complex sialylated, and there's a lot of those. Okay, that's really nice. So what we were able to do then is actually look across each of the glycosylation sites. So here we have 14 sites that we have in GlyConnect that are in common with the sites that are found in UniProt. So what's interesting here is that UniProt actually assigns a glycosylation type if it knows about the glycans that are found. And we found that, for example, here, this site, 122, in our, in the overall kind of look of this site, we find it as more complex, whereas they find it hybrid, which is kind of interesting. And then what was more interesting was that actually in our data set, we found all of the proteins, I should have said that earlier, they're all recombinant, okay, these COVID proteins. And so, oh, sorry. There we go. On the left here, we have ones, these proteins were actually across the entire S1 subunit. These ones here were just the receptor binding domain. So you can see in the receptor binding domain, there's actually only 2 glycosylation sites in the amino acid residue sequence. So what's nice here is that you see these hex cells, and these CHO cells all have very similar, if we just look at the two sites that are common to 3, very similar glycosylation, but this expression system was an insect cell line and it's quite different, okay? So the next step then was going back to epitopes, because that's one of the reasons we started this actually. So I took there's a published list of human epitopes from, I think it's 2009, and it's a really well-known one in the world of glyco, and it's got 118 epitopes that are found in humans. We created the glyco-CT for all of these. These are the list here. And taking that 114 list of epitopes, we queried, using GlycoQL to create the patterns for all of the epitopes, we queried GlyConnect, which has over 5,000 structures. And what we generated was a mapping between our structures and this list of epitopes. So for example, if I go back to the lactosamine again that I spoke about earlier, this is the mapping, this is the mapping for the lactosamine, okay? And each of these is an ID for a structure in GlyConnect. So we were able to use that mapping. We've stored it in GlyConnect as well as in the triple store. And we also use it to back up our conceptual map tool, which is Octopus. So you can scroll here and look for particular epitopes. So the next step was actually generating a user interface that users could use. So here we have, it's a very simple interface at the moment. You enter a glyco-CT string, which is, again, if you remember the format behind the cartoon. There's a couple of options here that you can click. Then you click search. And what you're given is a list of structures, the IDs which connect to GlyConnect, and the structure that you were looking for. So the next step, this is actually ongoing at the moment. we're going to incorporate a drawer, because a lot of glycobiologists would be very used to this kind of drawer and they'd find it very easy, even more so than actually putting in a glyco-CT string. So we want to repeat this for other epitope and determinant sets. So for example, in unilectin, there are over 200 ligands that are associated with different lectins. So we'll take this list from unilectin, again, create glyco-CT, and then create our sparkle queries, and sparkle patterns, sorry. Blast these against our GlyConnect data of glycan structures, and again, create a mapping. And this will be our automatic link between GlyConnect and unilectin. And so, for example, if I go back to my first example, these are mannose residues. So the reason we thought this was a good idea is because a lot of the databases that are involved in glycobiology already have ontologies behind them. So for example, we have GlycoRDF, which describes experimental data. GlyConnect itself is based on the glycoco, or the glycoconjugate ontology, which describes metadata around glycan structures. So now we've added to this the GlyStream ontology, which kind of gives another layer on top of these ontologies, and we are in the process of creating an ontology for unilectin. So in the future, we are going to look at repetitions, which are really important for GAGs, or glycosaminoglycans. They're repeating units, not as easy to represent. And also, I should have mentioned earlier, I spoke about the fact that we have fully defined structures. So out of our over 5,000 structures, only about 1,000 of them have fully annotated structures. But with these 1,000, we can give them KEBI identifiers. And using these, we can link to reaction databases such as REA. This is our group. This is my boss here, Frederic. And I just want to thank all of them and our funding bodies, and I'll take questions. Thank you.
Thank you for the talk.
No
It's
problem.
always interesting to see that apart of proteins there is something else attached. We have time for a couple of questions. If not, I will do my questions. That will be forever. No questions? No one wants to learn Sparkle?
Thank you. Bit far.
Hello.
Hello. I just wanted to, since there are so many ontologies describing different things, and you were saying like the next steps would be, for instance, to connect with reactions and more information. Like, what is the plan? Like, to create more and more ontologies, or like at a certain point to like unify everything in one big project? Yeah.
Well, I mean, the thing is that if we create ontologies, you're backing up the semantic web, you're creating links that anybody can actually link into. Now, at the moment, so to create some of this data, I did what are called federated queries between GlyConnect, the GlyStream, and UniProt. So instead of having to go across, if you imagine just a simple example, exactly what I showed, instead of having to go between the three, you can create one query that will do all of that for you.
Okay.
And that's, now at the moment, federation is a huge, in Sparkle, it's a huge question because it can be quite slow, but I'm guessing that in a few years they'll find ways to make it faster, and there's ways of kind of improving your queries to make them faster. But it's a really powerful thing to do, to be able to do. So that's the long-term plan, yeah.
Okay, thank you.
No problem.
Well, for that, all these ontologies should be stable enough just to be there when you are finding them.
Well, this is it. And also, I mean, I think what I've learned from this is that some ontologies they try to describe too much. So I think with our GlyStream ontology, I won't say it's simple, but it's not too complex, and it describes one aspect, and it allows us to look at the glycan structure in a very simple way, but also then the federated queries to GlyConnect allows us to add the metadata, which is really important, because the glycan structures on their own are They're just pretty cartoons.
Okay. There's no more questions, I think. I had a quick one, more in the structural side. How degenerate are these sugars, these glycans? Because there is many of that have different. It's not
Yeah.
is
You
it
mean
just a
how-
problem for the experiment, or is because they are actually degenerate structures? They are mixed structures.
How do they deduce the structures, is it?
No, I mean, so in a given glycan, you see that some of the there is uncertainty in the experiment, the same which is actual
Yeah.
structure. But my point is the actual structure is fixed, is a well-defined one, or is because you have also some changes in
Both. Both. So, I mean, on the one hand, glycans are not template-driven. It's to do with what enzymes are present, what the environment is like, and so that's why we have a number of different, if you like, scaffolds that present epitopes. But they are defined structures at the same time. The reason we don't have more defined structures is because they're branched, they have different isomers, and so they're not as straightforward as DNA or protein to deduce the structure. So there is usually a whole host of ways of deducing the structure of a glycan. So mass spectrometry, UPLC, and digestion. And so you don't always get all of the information. So, and also you have to use biosynthetic, what you know about the biosynthetic pathway. So we don't always have all of the information. Actually, in this case, older papers are great because a lot of the older papers have NMR data, which is the holy grail when it comes to structure representation. So
Easy field. Thank you very much.
no problem. Thank you.
For the talk. And now our last speaker is Peter Smirnov, if I remember properly. I think complete the talk he gave yesterday with an hour or two, orchestrating and sharing large multimodal data from transparent and reproducible research.
Yeah. Okay, thank you. So, as I was introduced, my name is Peter Smirnov. I'm a PhD candidate at the University of Toronto, and today I'm presenting our data sharing and pipeline orchestration platform, which we call Orchestra. This work was led by a master's student who's since graduated our group, Anthony Mamaliti, and it's now led by Minoru Nakano, who's a software developer in our group. Unfortunately, he couldn't be here today, so hopefully I can do their work justice. So the motivation behind this project, there's been a lot made of the reproducibility crisis in research, and really I think that computational biology and bioinformatics, in a way, we're uniquely situated to not contribute to the problem. A lot of what we do is we take data and we analyze it with code that should run the same today as it'll run 2 years from now. But we know that it's not that easy. And you know, working with published data and recreating results is still hard. And the way we see this, there's two aspects to this problem. One is getting methods to work that have, let's say, have been published, were used in the study, and but getting them to work in your hands is sometimes difficult. But the second aspect, and what we're trying to address here, is making sure that you get the data right and that you're using the same data, or the data that you expect to be using. And I think there's many aspects that are currently contributing to challenges working with biomedical data. First of all, we're increasingly taking one sample and profiling it with more and more technologies, and keeping all of these different views onto the same sample synchronized is sometimes difficult. Us computational scientists, we keep doing our work, we come up with better tools, and so the state of the art 2 years ago is different from the state of the art today. And more we see large ongoing studies which will release data as it's collected or in several batches, as well as they'll do quality control they'll look back at the data they've released before and maybe remove samples. And sometimes keeping track of that could be difficult. Now, some large, well-funded consortiums, they're getting pretty good at keeping track of their own data. And they can implement versioning schemes, but not everyone, first of all, not all studies have the same level of versioning. And second of all, when you try to integrate between, let's say, a large data set like TCGA and a smaller study, it could be difficult to keep everything in sync. And also, data is often going to be available with only one canonical pipeline. And if you're, again, trying to analyze across 2 different studies, if the authors didn't agree on what the canonical pipeline should be, you're introducing a technical batch effect that, theoretically doesn't need to be there. And for those of you who are at my talk yesterday, what we're motivated by is cell line pharmacogenomics data, where it's very multimodal. We have many different omics type, as well as drug response in cell lines. And this is one of those example of data sets that get continuously updated, and we have to keep track of versions of data. And so the question we're trying to answer is, how can we process and share such complex data sets using many different pipelines in a way that it will be reproducible and also keep track of the provenance of any data object that we create. And the solution we've come up with, we called Orchestra. So it's a cloud-based platform. It's supposed to help us achieve transparent, reproducible, and flexible analysis and also sharing of biomedical data. We started with this pharmacogenomics application, and it's helping us keep track of the different data layers while automatically versioning all the data and all the processing that we do. And we build upon an open source project called Pachyderm. And importantly, it also allows users to ask us for data sets processed with their pipelines and with their favorite reference. And it creates fully documented objects, basically with your choice of processing technique. So it's built in 3 different layers. There's a web app layer that users actually interface with. There's a data processing and orchestration layer behind that. And then for data sharing, we use the excellent Zenodo project as the repository for our data. Everything gets assigned a DOI, and then we, back in our web app, provide metadata on top of these objects and the documentation that's required, together with that DOI. So going through a bit about these 3 layers, the web app layer, what it would look like if you go to orchestra.ca, there's a front page, which, as I'll talk a bit later in the talk, we're looking at different data types, but for now, I'll focus on pharmacogenomics, where sort of our platform's most advanced. So if I click into there, you have a screen which allows you to either search for existing objects or request a new object to be created. There's our canonical processing choices are available, so if you don't want to make decisions. And then there's just some statistics about what's being used and how many, basically, if we have multiple processing queued up, it gives you an idea that you may have to wait for your object to be created. So if I go into the search and request function, here there's basically 2 things that happen. On the right side here, you can select different parameters for a particular, so you can choose a data set that you want to download, and then you can select parameters such as which molecular data levels you want included in your download, what version, in this case, of drug sensitivity data you want with the object, and, for example, which reference genome or transcriptome you want to use, and which, for example, tool for RNA quantification you want to be used on the data. Now, if an object exists, if it has been created already on this platform, it'll just search our database, it'll find the DOI for those particular settings, and it'll provide the object for you right away. If not, then we'll create one basically on demand. And the way we do this is in the data processing layer. So the request comes into our database. This tool, Pachyderm, which I'll describe a bit more on the next slide, it's constantly watching for new commits to the database, and it orchestrates the running of pipelines basically on the cloud using Kubernetes. Once this object is created, it's going to be kept track of inside our database, as well as kept track of on Zenodo, where it's assigned a DOI. That DOI is fed back to our web app. The user is emailed that their object is ready, and it's then going to be available for future users of our platform. So what is Pachyderm? Pachyderm is a system, it's an open source project, as I mentioned. It automates the execution of pipelines, but importantly, it does this together with data version control. So it does a very good job of connecting commits of code with basically commits of data. It tries to fuse basically a Git for code and data together. And as you develop new scripts or you have new versions of data, you can go back in history and see what code was used to create each object in your repositories. And importantly, whenever there's a change, if there's a change to a script, it'll automatically update anything downstream of that. And if there's a change, for example, to the raw data, it'll reprocess just the files that have been added, keeping track of the dependencies. Obviously, you need to configure the pipelines, but once it's working, it's almost magical when it works. Unfortunately, there's some drawbacks. Nothing is perfect. For us, the two main challenges we face is it's really made to be cloud native, and it works really well in platforms like AWS or Azure, but that has been or can get expensive quite quickly for academic researchers. And additionally, it relies on their own PFS file system, which powers the data versioning. But basically that means whenever you want to fetch an object, you have to translate from their proprietary or from their own file system to something more standard. So the way that we work around this is we've built a system to offload the very intensive computational processes to our on-premises HPC. And basically we do the usually first few steps that are very data intensive on premises. We upload that data to the cloud, where some of the lighter weight pipelines are also run by Pachyderm. And as soon as the process data is in Pachyderm, it again starts getting versioned. And then all the assembly of different combinations of objects is done in the cloud. And to keep track of what was done on premises in HPC, we use a biocompute object together with the pipelines run in Pachyderm, and that gets uploaded to Zenodo together with the data. So finally, the data sharing layer. I'll go through this briefly. So for an example, once you request an object, it gets assembled by Orchestra. It'll create automatically metadata web pages. So here you'll see there's links to the DOIs for the biocompute and the object itself. And also importantly, we make sure whatever data that we provide, if it's from a public study, we try to have the data usage policy, so you know basically what you're allowed to do with this data set and the citations that the authors ask you to cite. And then for different molecular data types, you get information about basically the annotations that were used to process them, as well as which tool, in this case it was Callisto, and the GenCode v33 transcriptome. You can look at a JSON representation of the pipeline that was run, as well as we link out to all the pipelines as they're stored in Git. And finally, we also automatically, for particular data versions, create release notes. So you can see for this particular version how much data was added, whether there's any new samples, how many experiments may have been removed. And this has really helped us stay sane when we ask the question, why did our results change? We can realize that there has been differences, or some experiments that we were expecting may have been removed. So finally, I want to end with the fact that we're really trying to push this beyond just our comfort zone of pharmacogenomics. We're working with other pharmacogenomic type data sets, radiogenomics, where basically it's cell lines treated with radiation, some toxicology data sets that look at gene expression changes after exposure to toxins. And right now we're particularly focusing on collecting and reprocessing immune checkpoint blockade inhibitor studies. It's obviously a very hot area of research in cancer, and there's a lot of data sets that are being published, but again, processed with different pipelines and not necessarily easy to do meta-analysis on right away. So finally, I want to thank everyone in the lab, obviously, especially for this, our HPC core at the University Health Network, our funding agencies, and there is a highlight talk, so there is a paper associated with it at that QR code. And I want to thank you for your attention.
Thank you, Peter, for the talk. Very nice. And we have time for a couple of questions. Yep, that's one.
Thank you for the talk. It's a pressing problem. You talked about avoiding the cloud because of computation costs, but the secret way they catch you is the data storage costs. But you seem to rely on the data storage to trigger pachyderm. Have you really just not avoided the problem?
Yes, so that's a good question. I think data storage has been manageable in costs. I think it's, for us, where they've sometimes caught us is getting data back off the cloud, and especially with larger data sets. So why I think this system works is we do sometimes upload large files onto the cloud, but usually we're downloading only the processed version of the data, which is smaller in size. But yeah, it's we're currently working on seeing whether we can replace, basically the pachyderm is an existing tool. We're trying to see whether we can stitch together something from a couple of other tools, something like a pipeline orchestration platform like SnakeMake, together with a project called DBC for data version control, whether we can recreate the same thing in a more portable tool, basically.
I will recommend WiFX. That is a product of the EOS part that to produce arrow crates that is very much similar to your final object, so it's called. So it's more or less these things. And all about this. More questions?
Yes, thank you. I think it's an excellent tool and platform. The question is, can you deal with data that has restriction in terms of what you can share? Like maybe you can share the downstream results, but not the data itself. So is there a way to control that and to expose what is only meant to be exposed, or even to have data sets that are like closed and you need to request access to just see it?
Yeah, so that's a very good question. We're developing tools to work with that right now, but they're not, I wouldn't trust them yet. And they're not, like, before we can release them for our own use, even at our hospital, they'd have to go through all the security, certifications by our security group. So maybe once that's done, we'll be more comfortable releasing that to the public. But yeah, it's definitely, especially with immune checkpoint inhibitor data sets, that's a relevant question.
Thank you.
I have time for a really, really quick one.
Yeah, maybe me. Yeah, hi. So excellent work. I think things are moving in the right direction in terms of reproducibility, which I think is something that we really need. I have a couple of questions. One based on, if you guys are also discussing the long-term strategies about how to maintain this, and what is the business essentially model that how you plan to use to maintain this long-term. And the other question is more about a comment or your opinion about, I believe that, a data set, a repository, data lake is as reproducible as all the steps that you do before to generate essentially this data. So we know also that, for example, sample storage, sample preparation, all these kind of things might be quite reproducible. So are you guys also trying to initiate a discussion with what is happening before essentially the data generation and the data storage to, for example, reach out to people or to labs or to facilities that you can have a little bit more guarantee that they have protocols, SOPs that generate standardized things, or you are more in line to ingest all sorts of data and just take care from the point of the data generation on? It's like, what's your point of view on this essentially?
Yeah, so I'll answer the second question first. We're leaning more towards ingesting what exists. And I think where there's opportunity is to make sure at least that all the relevant parameters are shared. So at least we know how the data was generated, even if let's say SOPs are not going to be stable over time. And honestly, with some of these cell line data sets, I think when we started working on this project, between then and now, definitely studies have gotten a lot better at sharing the relevant experimental parameters, such as even what media the cell line was grown in. In terms of long-term support, right now, actually, I can go back. This project was funded in Canada by a Genome Canada grant. Unfortunately, grants for building such platforms were not renewed in Canada. So I think it's a struggle for us just as anywhere. And I think we're lucky to have good institutional support that basically supports my colleagues that are software developers developing the software. And I don't know whether...
Are you guys looking also for private fundings in terms of support? Are you open to these possibilities?
So we've looked into this, but I don't think we've quite figured out how to monetize it while still keeping it in line with sort of our motivation and also our research interests.
Thank you. Appreciate it.
Thank you.
Well, this comes to an end. So thank you very much, the three speakers, for wonderful talks, and we'll find the next section.